So in this talk, I'm going to talk about some science that we've done with these probes as a sort of illustration project of the kind of scale that you can sort of think about bringing to your experiments using Neuropixels probes. Matteo, do please give me a 5-minute warning because I don't think I'll be able to keep track of it usefully. Okay. So I just want to super briefly set some background with this little video. So here you're seeing a mammal out in the wild doing a mammalian behavior that we would call in the lab a perceptual decision. Specifically, you can see this deer is trying to see if he can detect a particular sensory stimulus, an auditory or a visual stimulus, and discriminate what is the nature of that stimulus so that he can make a decision about whether to run or to attack or to ignore. And so in this project, we were trying to ask these sort of basic questions about perception that we still don't have good answers for in terms of neural mechanisms, specifically detection. Why are some stimuli perceived and others are not, even if it's an identical stimulus? And discrimination, how are multiple pieces of sensory information combined to make a decision about the nature of the sensory stimulus? And our work here really was inspired by some sort of heroic and classic studies. In particular, most inspiring to me, these studies from the Roma lab, where they recorded from many different cortical areas, from primary somatosensory through areas of frontal cortex and secondary motor cortex, all the way to primary motor cortex, looking for distinguishing features that would sort of spell out a pathway from sensation to action along these cortical areas. And in fact, what they found in these classic studies was that in cortex, this is not really true. That I'm not going to, suffice it to say, these plots show the types of coding that are observed in these different areas. Green means you code one sensory stimulus, red means there were neurons coding a second sensory stimulus, and blue means neurons coded the difference between the two, which was the relevant variable in this task. And the point here is that except for S1, which has only red and green neurons, every single other area had essentially the same combination of different signals. In other words, it was not the case that these aspects, sensation, decision-making, and action selection, action execution, were distinguished in different processing nodes within cortex in this particular task. And even M1 continues to have these red and green neurons all the way at the end of the trial. That is ones that represent stimulus 1 and stimulus 2. And so in this project, we were really asking, wanting to take this sort of concept and look actually beyond cortex and to see whether we could in fact identify a sensory motor processing chain if we looked more broadly in the chain, and if not, to characterize where we see these kinds of variables that relate to the performance of a behavioral task across the brain. Let me skip this slide because I'm a long time. So the the task that we designed, led by Chris Burgess, who's now at DeepMind from Matteo's lab, is a visual contrast detection and discrimination task for mice. You start with a mouse who's surrounded by 3 computer screens on which he'll see visual stimuli. The mouse uses his 4 limbs to turn a rubber wheel clockwise and counterclockwise. We reward the mice with water to train them which turn of the wheel is the correct answer in the task. And the mice are head fixed, which allows us both to precisely control the location of the visual stimuli relative to the mouse, and also, of course, to combine this behavioral task with the Neuropixels recordings that we did. So what do they see on the screens? They see a stimulus either on the left screen or the right screen. If it's on the left, they should turn the wheel clockwise. On the right, they should turn the wheel counterclockwise. And when they turn the wheel, actually, the stimulus will move on the screen. So another way to say what they're doing here is if it shows up on the left, they need to turn the wheel such that the stimulus moves to the center. If it shows up on the right, same thing, but the wheel has to move in the opposite direction. In some trials, there can be 2 stimuli at once. This makes it a contrast discrimination task. So they've got to compare the contrast on the left and the right and pick the one with higher contrast. And on other trials, there can be no stimulus presented. The mouse can still earn a reward on these trials, but only by holding the wheel still for 1.5 seconds. The mice can do this task very well. Here's a sort of psychometric curve showing that when the stimuli are on the right at different contrast levels, low, medium, and high contrast, the mice frequently choose turn the wheel such that they're indicating that they see a stimulus on the right. When it's on the left, they do the opposite. And when there's no stimulus presented, they're frequently making this no-go option, which they rarely make, only sometimes for low contrast stimuli otherwise. And so you can see that the mice are performing this task in a sort of, and it's quantifiable in a way that psychophysics tasks have been in humans and non-human primates in the past. And in particular, here's the sort of full data set as a function of the left and right contrast. Sorry, let me just explain that. Function of the left and right contrast. So you can see that mice are indeed making the no-go response when it was required, when both stimuli are 0, and they're trading off left and right choices as a function of the difficulty of the discrimination. So then, as you can imagine, what we did here was to record with Neuropixels probes during performance of this task. So here you can see a video of the mouse with the Neuropixels probe inserted doing the task. This is the screen that the mouse sees. So you can see a stimulus comes up, mouse gets a go cue and slides the stimulus towards the center of the screen by pushing on this wheel. Hopefully the task is reasonably clear. And in this example recording, we had two probes. The first one you can see up here going through secondary motor cortex prelimbic and secondary motor cortex anterior cingulate and prelimbic, and this one down here going through primary visual, parts of hippocampus and parts of thalamus. And so you can see at a glance that there's lots of interesting dynamics across and within these different areas. And you can see that we're really able to collect data from many different areas and many different neurons simultaneously. And here's a summary of the places that we recorded during this series of experiments. So we used 10 mice, had 39 recording sessions, and in total recorded about 30,000 neurons from each of these recording sites. So each colored line that you see here is a 4 millimeter stretch that describes the that shows where recording sites, the 3 to 4 recording sites, were located for one particular probe in one particular recording session. Okay, so let me just step aside from my experiment super briefly to say that our lab, and me in particular, are not the only people who can achieve this kind of recording scale, really recording neurons across the brain with Neuropixels probes. In fact, one of our students at the very first edition of this course in 2017 went on to do the very similar experimental design recording with Neuropixels probes from across the brain, and had a science paper just last year on this. So I think if you want to achieve this kind of scale in your own recordings, it's clear that this is a replicable procedure to do that. And I'm sort of proud to also call out here a couple of other papers that came from attendees of our first year course. I really think that this is a technique that you can bring back and immediately implement in your own labs. Okay, so more detail on these recordings that we made. So we recorded neurons in each of 42 different brain regions that I'm outlining here. These included primary and secondary visual cortex, frontal cortical areas like prelimbic and secondary motor cortex, primary motor cortex, somatosensory cortex. It also included subcortical areas, including parts of the basal ganglia in purple from the striatum, which is called CP, the caudoputamen, that's the input structure of the basal ganglia, to the substantia nigra pars reticulata, one of the output structures of the basal ganglia. It included parts of the thalamus, including visual and non-visual thalamic nuclei. It included parts of the midbrain, including the superior colliculus, classically known to be involved to receive projections from the retina and be involved in generating motor outputs, as well as some understudied areas like the midbrain reticular nucleus and the zona incerta. So what did we see when we made these recordings in this behavioral task? So I'm going to start with an example neuron just to sort of show you the kind of data that we're working with. So this neuron was localized to primary visual cortex. It had this waveform on the probe. So each one of these traces represents the voltage that we saw at one particular recording site located on one of the probe. And this was the activity of this neuron during the task. So these are spike rasters showing the spike times of a neuron on each individual trial, sorted by the contrast, high, medium, or low contrast for the contralateral stimulus, the stimulus on the other the screen opposite the hemisphere that this neuron was recorded in. And these are the average activity across those types of trials. So you can see that this visual cortex neuron is responding robustly following stimulus onset. And that's not surprising. We show a visual stimulus, you'd expect a neuron in visual cortex to respond. How about the rest of the brain? Do neurons elsewhere also respond after you show a visual stimulus in this task? So to visualize this, I'm going to take the activity of the neuron, which is a trace that starts out low and then increases over time, and turn it into a row in a color map. So it starts out white and then it turns to black over time. And so now each row in this color map is one neuron. So here we're visualizing some hundreds of neurons from primary visual cortex, as well as hundreds of neurons from secondary visual cortical areas. And you can see not just primary visual cortex, but also neurons in secondary visual cortical areas also respond following visual stimulus onset during this task. So now we get to zoom out and see across the brain. And in fact, what we were initially very surprised to observe was that in essentially every region we recorded, from frontal cortex to hippocampus to nuclei of the basal ganglia, nuclei of the thalamus, and all of these midbrain structures, we observed neurons that indeed responded following visual stimulus in this task. So it is not the case that responses are limited to a subset of areas in the brain. Instead, the entire brain seems to have changes in its responses during performance of this behavioral task. Okay, so how can we determine what about the behavioral task is driving the responses of these neurons? So to do that, we take advantage of the fact that we have these multiple different trial types that allow us to discriminate properties of the neuron responses. So here's an example neuron in visual area PM, one of the secondary visual cortical areas. And this neuron, you can see, has a response following visual stimulus onset at time 0 here on contralateral stimulus trials. This is a left visual cortex neuron, so trials when the stimulus was on the right, and not when the stimulus was on the left. And that's true whether the mouse correctly detected and responded to that stimulus, as in these trials, or whether the mouse failed to detect and respond to those stimuli, as in these trials, or even whether we just presented the same stimuli later during the day, after the mouse was already done with the behavioral task and was no longer doing any kind of active behavioral responses to any stimuli. And that's shown in these trials. And so this property of having a visual response that does not depend on the behavioral outcome, we would call a visual response, unsurprisingly. And we found such visual encoding in what is largely known as a sort of classic visual pathway. In other words, we saw in primary visual cortex and secondary visual cortical areas. We saw in the superficial layers of the colliculus, which receives direct inputs from the retina. We saw this in visual thalamic nuclei. And then we saw it in some areas that received direct inputs from primary visual cortex, such as these frontal cortical areas here and the striatum. Interestingly, we also saw this in some midbrain nuclei, and of course, we'll come back to those midbrain nuclei in some later slides. So what other kind of responses did we observe? So here's a different neuron. This is in the subiculum region of the hippocampal formation. And this neuron, you can see, it responds on trials with correct choices and not on miss or passive trials. So it's not responding to the visual stimulus per se. And in these plots, you can see that you can see these black dots. These black dots indicate the time of movement onset in the task, the time when the mouse begins turning the wheel. And down here, I'm actually just aligning to the time of movement onset. And now the black dots represent the stimulus onset. So we can see that, in fact, this neuron responds leading up to the time of movement onset. It responds starting before movement onset for both contralateral and ipsilateral choices, that is clockwise and counterclockwise turns of the wheel. So we would call this an action responsive neuron. It responds around the time of action, leading up to the time of action, but in a way that's non-specific to the particular choice that the mouse makes, contra versus ipsi. And we found this kind of encoding globally. Essentially, every brain region that we recorded, there were at least some neurons that had this kind of encoding. And these neurons really account for the majority of what we observed on the initial slide, where I showed you the neurons responding in many different brain regions during the task. And I think this is a really important thing to realize if you are going to make recordings across many brain regions yourself is that when the mouse executes a behavior, and probably any kind of behavior, you are very likely to find neurons that correlate with the timing and potentially with the magnitude of those behaviors. And so it's really important to sort of like recognize that itself does not constitute a specific signature of the particular cognitive process or behavior that you're trying to study. It's instead related to this global action encoding, non-specific action encoding that we see across the brain. Okay, so the next thing we wanted to consider was whether we could answer one of these questions that we started out with at the beginning of the talk, which was, why are some stimuli perceived and others not perceived? And so we tried to see whether we could observe a difference between conditions when we show the stimulus and the mouse responds to the stimulus versus when we show the stimulus and the mouse does not respond to the stimulus. And so here, the first thing I'm showing is the difference between trials during the behavioral task in blue and trials when the same stimuli are just passively presented in gray. And so these particular trials from the task are missed trials when the mouse did not respond. But in general, this in the blue traces, the mouse is engaged in the task and performing the task. Whereas in the gray traces, the mouse is completely passive and just observing the stimuli passively. And we observed that there's actually a big difference in the pre-trial baseline firing rate in many brain regions between when the mouse is engaged in the task versus passively observing the stimuli. And in fact, this pattern of differences in firing rate was really consistent across major divisions of the brain. Specifically, we observed decreases in firing rate in cortex. So the blue color here indicates that firing rate was lower during the task than during passive presentation, as you see for these two cortical areas here, with a small effect for this frontal cortical area as well. That's why it's a lighter blue here. But for subcortical areas, the opposite was true. We have here an increase in firing rate for the striatum, an increase in firing rate for these midbrain nuclei, an increase in firing rate for the CA1. And that's indicated by these red colors on the lower part of this plot here. And a prominent exception is these visual thalamic nuclei here, which instead follow the pattern of cortex. And so this was really striking to us, and it suggested to us a sort of explanation for what happens in the brain when the mouse is engaged versus disengaged in the task. And specifically, the explanation is the subcortical areas ramp up their activity during engagement, bringing the activity in those nuclei closer to a threshold for eliciting a behavior. And so to test that idea, what we wanted to do was ask, is this difference in firing rate pre-trial consistent with the difference in firing rate between miss and hit trials during the task? So in other words, if we have some difference between passive and task, do we have the same kind of difference between correct detections and misses? If this difference is really about the ability to detect the stimuli, then we ought to see basically even more of a suppression in visual cortex and even more of an enhancement in subcortical areas on those hit trials in the task. And so the scatter plot is showing exactly that. So the change in pre-stimulus firing rate, task versus passive, relative to change in pre-stimulus firing rate, go versus miss. And you can see there's a positive correlation across the brain in these parameters, thus allowing us to predict the probability that the mouse will actually make this successful detection just from using an index defined based on the pre-stimulus activity. Let me skip these control plots and give you this cartoon to help you understand how we're thinking about this result. What we're thinking is that there's a threshold, which in this hypothesis is likely to be defined subcortically somewhere, for instance, perhaps in the midbrain. And when you have a pre-stimulus state that's suppressed in key areas, such as these subcortical areas, then you're far from the threshold, and an input is not sufficient to drive you above the threshold. Instead, you approach the threshold but return back to the baseline activity, failing to generate a response. Whereas if you're in the task, or especially if you're in the task on trials with a go bias, your baseline activity, your pre-stimulus activity is close to the threshold already, such that even a small input is enough to drive you over the threshold and generate a behavior. So we think this is suggestive, requiring for the causal test, but suggestive of a mechanism for why some stimuli would be detected and some would be, and the mouse would sometimes fail to detect stimuli. All right, so what about, so I talked earlier about neurons that encode the action non-specifically, but what about this discrimination? What about how the brain actually determines which choice to make of the two? Are there neurons that instead do encode the time of action onset, but also the identity of the choice? The answer is yes, we found such neurons, but we found them very rarely. And I'll show you a few examples. So here's a neuron in the motor, the intermediate layers of the superior colliculus. That's its waveform. And this is a spike raster across contralateral choice, ipsilateral choice, and no-go trials aligned to the movement onset. And you can see this neuron responds more for contralateral than for ipsilateral trials, even sort of suppressed down to 0 for the ipsilateral trials. Here's another neuron in secondary motor cortex and the frontal cortex that is enhanced for the blue trials, the IPSI trials, one in striatum, one in the midbrain reticular nucleus, and finally one in the zona incerta. And so we found such neurons that discriminated left versus right choices in a diversity of brain regions, but rarely in each of these brain regions. And just here's sort of an example of one of these neurons showing these same plots that I showed you before. So you can see this neuron really strongly responds to the one choice, but not the others. And that's specific to the correct choice trials, not the misses or the passive presentation. All right, and so we found these neurons in multiple different brain regions, and I'm showing you a few more examples to illustrate the principles that we observed that governed what kind of signatures we saw in different areas. And specifically, what we observed is that in forebrain, that is cortex and basal ganglia, we observed bilateral encoding. But in the midbrain, such as colliculus and midbrain reticular nucleus, we observed what we call unilateral encoding. So specifically what that means is that nearly every neuron in the midbrain, which had this choice selective activity, preferred the contralateral choice. You can see all 4 of these neurons prefer orange over blue, whereas in the forebrain, instead, it was about 50-50 between neurons that prefer contralateral versus ipsilateral. And secondly, what it means is that in the midbrain neurons, we frequently observe this property that ipsilateral choices are suppressed below baseline, firing rates are suppressed below baseline for ipsilateral choices for the non-preferred choice, whereas in the forebrain, both choices have increased activity, just more for one than the other. And so that leads us to have this map of the choice encoding, which includes frontal cortex, primary and secondary motor cortical areas, parts of the basal ganglia and thalamus, and these forebrain areas have this bilateral encoding indicated by the brown color, as well as midbrain areas like colliculus, midbrain reticular nucleus, substantia nigra, and zona incerta that have this unilateral type encoding indicated by preference for one choice over the other and a suppressive interaction between choices. Okay, I think that's it. So the upshot here is that these Neuropixels CMOS probes really have enabled this unprecedented scale across the entire brain without loss of resolution. So we're still recording individual neurons, individual spikes. And they've enabled us and others to really tackle questions that we weren't able to even consider addressing before this technology existed. And so specifically in our study, they allowed us to identify a few principles that appear to organize the behavioral coding across the brain. Number 1, spontaneous and goal-directed actions are encoded globally in essentially every brain region we recorded from. And I didn't discuss it, but these representations in experiments with perturbations using optogenetics, experiments on which we have a preprint that I can point you to, we showed that many of these representations are not necessary for this visual task. Second, we observed that cortex is suppressed during engagement in this task, but subcortical areas are activated, suggesting a mechanism for why some stimuli are detected and others are missed. And finally, choice is encoded in midbrain unilaterally, but in forebrain bilaterally, suggesting that there's a distributed network that encodes the choice and that there's an inhibitory interaction potentially in the midbrain that discriminates between the two choices. So let me definitely thank my advisors in this work, Matteo and Kenneth. Peter Zakaas was a really talented graduate student who worked with me on these experiments, and especially on the optogenetic experiments I alluded to at the end. Thanks certainly to Tim and to everyone on the Neuropixels team for their work here. Happy to take any questions. That's all I have.
I think you were amply within time, Nick, so you're fine.
Great.
Let's see. We answered quite a few questions.
Okay.
As things were going. There's one that just arrived. Hi, Nick. At one point, you mentioned that during any behavior, if an experiment is recording from multiple brain regions, they will identify neurons with correlated responses to whichever behavior of interest. But that should not be interpreted as being behavior specific. But it could also be a result of propagating activity in global dynamics. Could you please expand a bit on this? And then the question goes on, how can one view activity in a given nucleus that correlates with a specific behavior? What experiment analysis should be done that provides sufficient evidence for the role of a given nucleus in driving or mediating a specific behavior?
Yeah, good. So I think the main point here is that you need to be sort of like thoughtful and careful in your behavioral task design. So if you have a go, no-go task where one choice is indicated by behavior and the other choice is indicated by no behavior, then this may be the kind of task that's difficult to discriminate what's a specific correlate of the cognitive process or the choice for that task, as opposed to this non-specific global thing. However, in our study, we had only a very slightly more specific, more complex design, right? But we had this two alternative forced choice where the mouse could turn the wheel one way or the other way, and that's what allowed us to observe that there were some neurons that only responded to one direction of wheel turn and not the other. And so I think having something in your task design where there's going to be a specific correlate that's not just possibly any action at all is really what you need to be able to discriminate.
Okay, yeah. There's another question about the science, and then there's a question about grounding. So let's do the one about science, and then maybe we could take the one about grounding. Both you and Carolina could take it, maybe. So the question about science is, were you able to observe any interesting oscillations across the regions?
Great question. So the one oscillation that we've studied in the context of this task was a project led by a graduate student in the lab, Alina Jacobs, and she was studying these low frequency oscillations that appear primarily in visual cortex, but also in other parts of cortex. They're around 3 to 6 hertz in the mouse. And it appears that those oscillations co-vary with the state of engagement in this task. She has a really nice paper using wide field imaging and also some electrophysiology to look carefully at the way that those oscillations and presumably the state of cortex co-vary with engagement in this task. And I think it's a really nice paper because this task allows discriminating engagement versus perceptual accuracy. And those oscillations do not co-vary with the perceptual accuracy, only with the engagement. So I would point to Alina's paper on that. I think there's a lot of room for analyzing other kinds of oscillations, like beta oscillations in the motor system, et cetera, but we really haven't gotten into that at this point. The data, by the way, are freely available, so please just check it out if you're interested. Okay,
so if you remember, Nick, after this, you may want to put in the chat the links to the papers you just mentioned. But now there's a question that is technical. I have a tetrode background using freely moving rats. I was taught that reference tetrodes comparable to internal reference are important to help with local noise not caught by the ground screw. Using your ground as reference was generally considered very lazy practice. Changing which reference tetrode I used could help with reducing noise. This was true when recording from multiple different brain areas at the same time. It is true that you can add by subtraction your reference signals into your recorded signals, but there are post hoc ways to clean that. The tendency I read in the Q&A here is to not use internal references except for motion artifact reasons. Is this a probe versus tetrode thing? Or maybe head fixed versus freely moving, or the result of new insights? Did you ever observe that using a local reference can reduce local noise?
I'm not sure about the very last question, using a local reference to reduce local noise, but I will say that I've been using the internal reference mode in my recordings recently. I do think it is easier to use and produces nice recordings. So I think if you're familiar with that and that one makes the most sense to you, go for it. I mean, I think that I'll talk a bit more about grounding and referencing tomorrow in the context of trying to give you some tips about how to like denoise your rig, which I'm not going to be able to tell you everything about it, but I can try to give you some ideas. And I think that, you know, in thinking about that kind of thing, like the fact that every electrical situation in every lab is different in some ways, I really think that the best strategy is going to be to try multiple different strategies for your setting and see which one produces the best recordings, because it may really differ just based on idiosyncratic factors that you can't control or aren't aware of.
Great. Let's see if Carolina wants to add anything. Carolina, I don't know if you're still around.
Yeah, here. I think that's correct. I think one issue with using very small internal electrodes, it has to do with the very high impedance, so this doesn't provide a good cancellation. But if you use a large internal electrode, I think that's the best for most of the experiments, except when you want to record LFP. I think for LFP, using external reference is always better, right? Otherwise, you will cancel a lot of your signal in the amplifier.
Great, thank you. I think the largest site is the one at the very bottom, the one at the bottom of the
exactly.
Yeah, and unless that's in a super interesting brain region that has crazy stuff that is picked up by a very large electrode, that would be a good reference to use.
Yep.
Yeah, and I guess I added this thought in text, but on one of the Q&As, but let me just add there though that I do think that, you know, there's really substantial volume transmission of LFP in the brain anyway. So even if you're using an external reference, you know, you want to think carefully about what LFP signal you're measuring and whether it's useful, which I think it is in most cases, to do local re-referencing of your LFP anyway, so that you're measuring a signal that's local to the sites that you're recording at. So current source density is an example of such an analysis that uses differences between sites to identify local LFP signatures, regardless of whether you did internal or external referencing. And I think that that's a good solution that dodges that problem.
Okay. Nick, there's 6 more minutes before the break, and there's two interesting, well, at least one for sure, new question. Let me read the other one later. From Aaron, he says, what tools are used to identify individual neurons that have an outweighed correlation with response? Is this done largely manually? That is, when you have 30,000 neurons, I suppose the question is, how do you figure out which ones have an outsized correlation with response?
Yeah, I can give a couple slides on that if you want. Do you think that that would be useful here? I guess we're 1 minute away from break, is that right?
No, we're 6 minutes away from break.
Oh, we're 6 minutes away from break. Okay, yeah, then let me just give a couple slides on that in that case, because I think that is a good question. Let's see, going back here. Sure. Okay. All right, so very first backup slide. So the way that we did this was with fitting a kind of model to the neural activity, which we're calling a kernel model, for reason I'll explain in a sec, that allowed us to quantify the magnitude of the contributions of each of these behavioral task factors to the spiking activity of each neuron. And the goal of this model is to explain the spiking of each neuron as a function of responses over time that differ in their magnitude and timing depending on which visual stimulus is shown and which behavioral response is given at the end of the trial. That is, whether there was a response at all and whether the choice was left or right. And so these are sort of example such kernels that give you the idea of what's happening. So if you remember that visual cortex neuron we looked at before, it had an early and high and large amplitude response for high contrast stimuli and a small and late response for low contrast stimuli. And so for such a neuron, you might have kernels that look like this, which have shapes matching those responses to the visual stimuli. And then the idea is that you can predict the activity of the neuron on any individual trial by taking a sum of the kernels that apply to that trial, shifted according to the timing of events in that trial. So specifically, like in this example trial number 1, the movement was early, in example trial number 2, the movement was late. And so the predicted spiking that you get is this black trace in trial 1 and this black trace and other black trace in trial 2, which differ because the timing of the events and the identity of the events was different. But in either case, the predicted activity just reflects the linear sum of the kernels that were fit to that neuron's activity. And so the idea, of course, here is to minimize the difference between these predictions and the actual spiking data by optimizing the shape of the kernels for each neuron. So how do you do that? So I will now briefly describe the method that we used to fit these kernels and to optimize that fit using the fact that we have a very large data set. And the way this works is that the kernels are fit with linear regression, very simply, and they're fit by designing a specific matrix that encodes the information about the trial events, and then simply doing linear regression between that matrix and your spiking activity. So the matrix is very carefully designed such that each row in this matrix corresponds to one predictor, which represents an event at a particular time offset. So this row here represents that a left high contrast stimulus is going to occur soon. This row here represents that a left high contrast stimulus occurs right now. And this row here represents that a left high contrast stimulus occurred sometime in the past. And the columns here are just time points during the entire behavioral session. And so then you simply fit this with linear regression. X, this task predictors, is this matrix here. And Y, the spike counts, is your spike counts over time of vector. And you get A, the mapping of regression weights from this predictor matrix onto your spike count. And then the kernel is just given by the weights for a given set of predictors over time. And you can think of that then as simply a mapping between X and Y with a set of weights indicated by these lines. Now, the difficulty with this method, and see the paper for like specific details on exactly how to assemble this matrix, and we've shared code online too. It takes a little bit of wrapping your head around it, but it's not in the end too complicated. So, but the difficulty with this method is that there are many, many predictors here. So this matrix has 650 rows, given just how we in particular designed it and what time bin size we used. And that leads to overfitting, or can lead to overfitting, depending on how much spiking data you have, how many trials you have. And a classic solution to try to solve this and reduce the problem of overfitting is by essentially encoding a smoothness prior in your predictor matrix. So you change the predictor matrix to have smoothness in each one of the predictors, and then you can use fewer predictors. So rather than say 90 rows here, you only have say 10 rows here, and that reduces the number of predictors by a lot, but forces them to be smooth, which if they're really smooth, then is a good simplification and a good sort of form of regularization to solve this problem of overfitting. What we tried to do here was something different. So we tried to take advantage of the fact that we have many, many neurons that can enable us to identify an optimal way to do that reduction in number of predictors. So specifically, we have the task predictors, which are 650 by time points, but then we have bin spike counts, and actually we have this from many neurons. So we have a matrix of bin spike counts, not just one neuron by time points, but actually many neurons, say 300 neurons by time points. And what we want to find is a low-dimensional representation of these task predictors that's optimally designed to enable predicting these spike counts of all these neurons. And this is what the predictor matrix looks like when you apply reduced rank regression, which is just a method from the linear algebra literature that does exactly what I described. And you can see that now these predictors are really, really different than before. It's a very dense matrix rather than a sparse one. But each of these rows is now optimally designed based on the data to predict the neurons. And then we can apply this method. I'm going to skip this slide. That's some details. And this slide is about estimating our false positive rate. But basically what we can do then is apply this method to get fit traces without overfitting to this spiking activity. And finally, to answer the question, once we have this model where we can fit the spiking activity of the neurons as a function of these different kernels, what we can do is leave one of the kernels out of the model and ask how much worse the model fit becomes without that kernel. So specifically for this example neuron from Visual Cortex that we looked at before, in the solid line, the thin line with shading is the data, the solid line is the full model, but the dotted line is the model if we leave out the visual kernels. And you can see this model is much, much worse. But if we look at the data for the same neuron aligned to the time of choice, We can leave out the choice kernel, we can leave out the action kernel, and actually the fit doesn't get much worse because all of the variance of this neuron really is explained by knowing the visual stimulus.
Let's stop here because we're over time now.